Thank you for joining this course. My name is Dr. Sky Bal, and in the coming course, we will be looking at the citric acid cycle, also known as the tricarboxylic acid cycle. Now, following on from our Metabolism 101 course, where we learned how pyruvate is generated from glycolysis and enters the mitochondria to join the citric acid cycle. Now, let's have a detailed course on the cycle itself. Firstly, a brief intro, the mitochondria. The mitochondria arose from prokaryotic endosymbiotic bacteria, which till now retains the remnant of its genome. Now, this mitochondrial genome is in a circular form and is within the mitochondria rather than the cell nucleus. Now, although the mitochondria has its own DNA, it still largely depends on the genes that were transferred to the nucleus of the host. In simple terms, many proteins playing critical role in mitochondria are coded by the nuclear DNA of the cell. Now, with the emergence of mitochondria in eukaryotic cells, it brought biochemical mechanisms that allow us to acquire and use energy more efficiently. So this is the figure from our previous course, the Metabolism 101 course, which is showing you how glucose is fed into the citric acid cycle. This glucose can be from the cell storage, which can be from glycogen, or it could be from the glucose absorbed directly. Another way of feeding into the citric acid cycle is through the beta oxidation from the fatty acids, which can also feed into the acetyl-CoA pool inside the mitochondria. And then that will join the citric acid cycle in the same manner as the acetyl-CoA from the glycolysis. Thirdly, there is glutamine that feeds into the citric acid cycle. And this is where glutamine is converted into alpha ketoglutarate, which then becomes part of this citric acid cycle. So we have 1, 2, and 3 main feeders into the citric acid cycle, the tricarboxylic acid cycle. These are the 3 main feeding points into the citric acid cycle that we need to remember. Interestingly, recently we found that lactate, which was thought to be a waste product from glycolysis, can also feed into the citric acid cycle. And this is done by converting lactate back into pyruvate, which then also again forms the pool of acetyl-CoA and hence joins the cycle. Okay, now let's focus on the citric acid cycle. In summary, the citric acid cycle contains 8 main steps, right? Which are condensation, isomerization of citrate, we have generation of carbon dioxide by an NAD plus linked enzyme. There is a second oxidative decarboxylation step. There is the substrate level phosphorylation step. There is the flavin dependent dehydrogenation. There is the hydration of a carbon to carbon double bond. And then finally, the dehydrogenation reaction that regenerates oxaloacetate. In the coming course, we are going to look into each of these steps and understand the biology and chemistry behind these reactions. So there's no need to get overwhelmed with the details shown at this point. Okay, so in the first step, acetyl-CoA, a 2-carbon compound, So the blue balls represent carbons that you see here. And as you can see, acetyl-CoA is a 2-carbon compound and oxaloacetate is a 4-carbon compound. Okay. So oxaloacetate and acetyl-CoA are converted to citrate, which is a 6-carbon compound. This reaction is catalyzed by citrate synthase. Now, citrate synthase has been conventionally designated as the first enzyme of the citric acid cycle, which performs the irreversible condensation of acetyl-CoA with oxaloacetate to form citrate. The rate of this reaction is controlled by ATP to ADP ratio, as well as the NADH to NAD plus ratio. For example, if the ratio of ATP to ADP is low, meaning that the cell requires more ATP, so there is less ATP, the triphosphate, then the rate of this reaction will increase and therefore produce more citrate from acetyl-CoA and oxaloacetate. Also, in this reaction, the coenzyme A is given off. Okay, so the coenzyme A from acetyl-CoA is taken off. Now, in another scenario where the energy demand of the cell is not high, the reaction may still occur, but then after this point, citrate will be transported out of mitochondria for other cellular processes. And this is done by mitochondrial citrate transport protein across the inner membrane. Okay? So the mitochondrial citrate transport protein transfer the citrate from the matrix to the space between the two membranes, the mitochondria. Thereafter, citrate can passively diffuse through the outer mitochondrial membrane. Remember, I said in the Metabolism 101 course that the citric acid cycle occurs in the matrix of the mitochondria, the space within the mitochondria. And mitochondria has two membranes, an inner and an outer. So citrate has to transport firstly from matrix to the space between the two membranes and then to the cytoplasm. Okay, so I strongly recommend that if you haven't watched our Metabolism 101 course yet, then please watch that first, as the present course will make much more sense if you have the background from the other course.